Imagine if learning to play the piano, or
Yeah.
even just craving your morning cup of coffee, literally, and I mean physically, warped the microscopic shape of your brain in real time.
Which is a crazy thing to visualize, but it's, exactly what's happening.
Right. And welcome to you listening to today's deep dive, where we are getting into exactly that.
Yeah.
Because for over a century, really since the legendary neuroscientist Santiago Ramon y Cajal first looked through a microscope, scientists have known the brain is essentially this massive, incredibly dense, impenetrable forest.
Yeah, forest of neurons. And for decades, the holy grail has basically been to map it.
And recently, we've gotten really good at mapping the DNA of the seeds in that forest, right?
Yeah.
What we call transcriptomes. We know what kind of trees are supposed to grow.
Exactly. We have the genetic blueprints. But
Yeah.
mapping the actual physical shape of the adult trees, like their exact branches, how they twist, where the tiny twigs end up, that has been a complete nightmare.
Like historically bad, right?
Oh, absolutely. Historically, researchers had to rely on century-old techniques. Things like Golgi stains, which are notoriously finicky, or they had to manually inject dyes into individual cells.
Which sounds exhausting.
It is. Tracing a single, complete neuron in 3 dimensions could take days of agonizing manual labor. You'd just be staring at a screen, clicking point by point, trying to follow a microscopic thread through this dense jungle of other threads.
Which is exactly why we are so excited to unpack the research for you today. We're looking at a groundbreaking 2025 study from Nature Neuroscience. It's titled Dendritome Mapping Reveals the Spatial Organization of Striatal Neuron Morphology.
And what these researchers did here is build a revolutionary new pipeline. They successfully mapped 3,762 complete neuron trees in the mouse brain.
Which, just to put that in perspective for you listening, in one single study, they literally doubled the entire historical archive of this specific type of neuron.
Yeah, it is a monumental leap in scale. And what makes it so important is that they didn't just map these neurons, they uncovered these highly specific morphological neighborhoods in the brain.
Right. They showed how the local zip code of a cell dictates its physical shape. And crucially, they looked at what happens to this physical structure during normal aging and in a model of Huntington's disease.
Which has huge implications for how we treat those diseases.
Okay, so let's start with the real estate. Where exactly in this vast forest are we looking?
So we are focusing on the mammalian striatum, and specifically a region called the caudate putamen, or CP.
Okay, CP. And if you're listening and wondering why you should care about this specific lump of tissue, you can think of it as the command center for your motor performance, your habit formation, and reward learning.
Right, it's the part of your brain that remembers how to ride a bike. And it's the exact same part that drives you to reach for a piece of chocolate after dinner without even thinking about it.
Guilty as charged. And the undisputed star of the show in this command center is a cell called the medium spiny neuron, or MSN.
Yeah, MSNs make up 90% of the neurons in the striatum. They are the absolute workhorses.
And from what I read in the sources, they generally fall into 2 broad categories based on their genetics, right? D1 type and D2 type.
That's right. So the D1 type MSNs form what's called the direct pathway in the brain. You can basically think of them as the go signals.
They initiate movement or action.
Exactly. And then the D2 type MSNs form the indirect pathway. They act as the stop signals, inhibiting action. The balance between those two is what allows you to move smoothly.
Okay, let's unpack this. If we already know they are D1 and D2, and we know their genetics perfectly dictate go or stop, why do we need to know their exact 3D shapes?
It's a great question.
I mean, why did these scientists spend so much time and money mapping the physical branches if the genetic code already tells us what the cell does?
Because in biology, form is function. A genetic label just tells you the cell's potential, but its physical shape tells you what it's actually doing.
Okay, how so?
Well, the branches of a neuron, which we call dendrites, act as the physical antennas of the cell. These MSN dendrites are responsible for integrating diverse electrical inputs from all over the brain, like the cortex, the thalamus, the midbrain.
So they're constantly catching signals.
Exactly. And their exact physical shape, like how long they are, how widely they branch out, the specific angle of their forks, that determines exactly which signals they can physically reach in the dense forest of the brain.
Oh, I see. So if a branch is just like 10 microns too short, it might completely miss a vital stream of dopamine coming from the midbrain.
Precisely. If you don't know the precise shape of the antenna, you don't truly know what radio station the cell is listening to. Yet before this study, the sheer physical difficulty of tracing them meant only about 3,700 MSN dendrites had ever been fully reconstructed in the entire history of neuroscience.
Wow. Okay, so let's do a quick recap here to make sure we're wrapping our heads around this. The striatum controls your habits. 90% of its cells are MSNs, which are your D1 and D2, your go and stop signals. And measuring their exact branching shape has historically just been too hard to do at scale.
Right, exactly.
So I want to throw a question to you listening. Think about a habit you have. Maybe your morning coffee routine. That's your MSNs firing. How do you imagine the physical antennas of those cells might physically change when that habit becomes deeply ingrained? Do they grow? Do they shrink?
It's a fascinating thing to picture.
It really is. Which brings us to the actual how. Because from what I understand, looking at brain tissue is basically like looking at a brick wall. It's opaque, it's dense, everything is tangled together.
Yeah.
How on earth do you take a picture of these antennas?
Yeah, that brings us to the technological marvel of this study. To map a forest this dense, you can't just look at it from above. They had to invent an entirely new systems biology approach, and it starts with incredibly sophisticated genetic labeling.
Okay, lay it on me.
They used genetically engineered mice called MORF3 mice, and they crossed them with CAMK2 a Krier and D1 TD tomato.
Wait, hold on. That is a serious genetic alphabet soup.
I know, it's a lot.
CAMK2 Krier, D1 TD tomato. What does crossing those specific genes actually do in plain English?
Fair question. Let's break it down. What this complex genetic trick does is allow for something called sparse labeling.
Sparse labeling. Okay.
Imagine looking at a dense, dark forest where every single tree is painted glowing green. It would just be a blinding wall of light, right? You couldn't trace a single branch.
Yeah, it'd just be a glowing blob.
Exactly. But this MORF3 and Krier system acts like a biological dimmer switch. By injecting a specific drug into the mice, they can essentially black out 99% of the forest and randomly light up just a few scattered D1 neurons in one neon color and D2 neurons in another.
So they isolate the subjects. They glow brightly, but they are spaced far enough apart that their branches don't overlap into a messy blur.
Precisely.
Yeah.
But there is another massive hurdle. Even if they light up, they are still buried deep inside the opaque brain.
Right.
Historically, researchers solved this by slicing the brain into incredibly thin sections, maybe 100 microns thick, just so light could pass through them.
But wait, if you take a massive branching tree and slice it into wafers, you're chopping the branches in half. You completely lose the 3D structure.
Which is why the researchers in the study used 500 micron thick brain slices. These are massive, chunky slabs of tissue, much thicker than the old 100 to 350 micron slices.
But a 500 micron slab of brain is completely opaque.
It is. It's full of lipids fats that scatter light. So they used a chemical clearing method called IDISCO plus A.
Okay, disco plus A. Sounds like a nightclub.
It does. But what this process does is literally wash away the fats while leaving the structural proteins intact. It replaces the water in the tissue with a specific chemical solution that matches the refractive index of the proteins.
Meaning it literally makes the brain tissue completely invisible. It turns a piece of biology into a block of clear glass, with these sparse, neon-glowing neurons perfectly suspended inside.
It's beautiful to look at. From there, they used a massive piece of hardware called a dragonfly confocal microscope to take high-resolution 3D photos of these transparent slabs.
Okay, so now we have the photos.
Yes, but you still have a data problem. Human eyes can't trace 3,700 complex 3D trees manually, so they trained a machine learning AI A2D UNet to hunt through these massive image files and identify the cell bodies, the somas, and the dendrites.
And I saw they used algorithms with names like GCUT and APP2. Stop there for a second. Are these just finding the edges of the cells? How does an algorithm know what's a branch and what's just background noise?
So these algorithms are specifically designed to follow the path of least resistance along a glowing signal. If two branches from different cells accidentally cross each other, the GCUT algorithm mathematically calculates the trajectory and angle of the branches.
To figure out which twig belongs to which tree?
Exactly. It automatically separates them.
Wow. Which brings us to the final step of the pipeline, which honestly might be the most impressive part. Registration. Because once you have this beautifully traced AI-generated 3D tree, you need to know exactly where it lived in the brain.
Right. A tree without coordinates is useless for studying neighborhoods, so they developed a novel, multi-scale registration algorithm. They used something called a computational anatomy random orbit model.
Okay, another mouthful.
It is. But what this highly complex math does is take these thick 3D tissue slabs and warp them just enough to map them perfectly onto a standardized 3D GPS map of the mouse brain.
A GPS map of the brain.
Yeah, it's known as the Allen Mouse Brain Common Coordinate Framework, or CCFV3.
So let's do another quick recap. If I'm trying to picture this whole pipeline, it's like taking a dense forest, using a genetic dimmer switch, that's the MORF3, to highlight just a few trees. Then you make the earth completely transparent with those 500-micron thick iDisco plus slices. You snap 3D photos, use an AI to trace every single branch, and then map that exact tree perfectly onto Google Earth, the CCFV3, so anyone in the world can find it, resulting in 3,762 perfectly mapped cells.
That's close, but the Google Earth analogy actually understates it because Google Earth is mostly a 2D surface projection. This is mapping down to the micron in three-dimensional coordinate space.
That is wild. So question for you listening. Which part of this pipeline blows your mind more? The biological trick of making brain tissue totally transparent, or the AI algorithm accurately tracing microscopic branches in 3D? Because both are just science fiction to me.
It really is a marvel.
Okay, so they have this massive, unprecedented database of perfectly mapped 3D trees. But how do you mathematically describe the shape of a tree so a computer can analyze it?
Well, you measure everything. They extracted 31 distinct morphometric features for every single cell.
31 features?
Yep. They used highly specialized software tools called LMeasure and the Trees toolbox, and they basically categorized these features into three main buckets.
Okay, what are the buckets?
First, length-related features. So the total length of all dendrites combined, or the segment length, the physical distance a branch reaches from the cell body. Second, angle-related features, measuring the exact angles where branches fork.
Right.
And third, complexity-related features. This includes the total number of branches, the number of tips, the number of primary stems.
They also measured something called centripetal bias, which I find fascinating. From the sources, it basically measures how much the branches are pulling inward toward the center of the cell versus reaching outward into new territory.
Exactly.
But why is a branch pulling inward a bad thing?
Because it creates a functional echo chamber. If a dendrite branches outward, it can sample neurotransmitters from entirely different pathways. If it pulls inward, it's only talking to its immediate local environment. It loses the ability to integrate long-distance signals.
Here's where it gets really interesting, folks. They have this massive spreadsheet of 31 features for every cell. What did they actually find when they compare the D1 go neurons to the D2 stop neurons? Did their genetic identity dictate their shape?
Well, yes and no. The overarching finding was that D1s generally have longer total dendritic lengths and straighter branches than D2s.
Okay, so genetics matters.
It does, but, and this is a massive but, this difference is highly dependent on their spatial zip code.
Their zip code.
Yeah. The striatum isn't just one uniform blob. You can break it down into subregions from front to back. You have the rostral at the front, or CPR, the intermediate in the middle, CPI, and the caudal at the back, CPC.
Wait, so you're telling me a D1 neuron in the front of the brain looks fundamentally different than a D1 neuron in the back? Doesn't that contradict the whole idea of genetic types dictating the cell's function?
It reveals that genetics are only half the story. The local environment, the zip code, exerts a massive evolutionary pressure on the cell's physical shape. The biggest structural differences between D1 and D2 cells were found in the intermediate and caudal communities. Down there, the D1s were distinctly longer and straighter. But surprisingly, if you look at the rostral area at the front of the striatum, the D1s and D2s were remarkably similar in length. Furthermore, across almost all the zip codes, the complexity features, the actual number of branches and tips, were largely indistinguishable between the two genetic types.
Let me summarize this real quick for everyone. They measured 31 shape features. D1s are generally longer than D2s, but these differences depend heavily on where they live in the striatum. CPI and CPC are very different, but in the CPR, they look the same.
You nailed it.
So if the physical shape of your neurons depends so much on their specific zip code in the brain, what does that tell you about how specialized our brain's internal neighborhoods really are? It implies that the local neighborhood is shaping the cell to do a highly specific job.
Exactly. And as the researchers realized, if the neighborhood is so important, the old ways we used to draw boundaries in the brain might be completely obsolete.
Which brings us to the next big breakthrough.
Right. Neuroanatomists have been drawing invisible borders on brain maps for 50 years, mostly based on crude cellular staining. But the researchers here wanted to group neurons based purely on their actual 3D geometry.
So what did they do?
They took their GPS map of the brain and overlaid a grid of 500-micron cubic boxes. They essentially chopped the virtual space into a massive grid.
But how do you compare a box with 50 different highly complex trees to another box? I mean, that's just too much data.
You need a summary. So they used a mathematical trick called WGCNA, or Weighted Gene Co-expression Network Analysis.
WGCNA, got it.
It's a tool originally developed in genetics to find groups of genes that turn on and off together. But here, they adapted it for 3D geometry. They created something they called the eigenmorph.
The eigenmorph.
Yeah. The eigenmorph is a single statistical summary shape representing the average dendritic features of all the neurons living inside that specific box.
I love this concept. So it's like trying to figure out the average vibe of a specific city block. You don't have time to interview every single resident about their fashion, their music taste, and their favorite coffee. Instead, you mathematically calculate a single summary vibe that accurately represents the whole block.
That's a great way to picture it, but the danger with the city block analogy is that a simple average might erase the really important outliers. What makes the WGCNA math trip different is that it preserves the complex, interrelated features.
Okay, how so?
It doesn't just average the length, it captures how length relates to branching angles within that specific box. And once they had an eigenmorph for every box, they asked the computer to group boxes together that had similar shapes.
And they completely ignored the old anatomical textbooks when they did this, right?
Completely. By clustering these boxes based purely on shape, they discovered 7 distinct dendritic modules. They called them morphological territories, or MT1 through MT7.
MT1 to MT7.
Right. These are contiguous physical swaths of brain tissue defined solely by the architecture of the cells within them. And what's fascinating here is that when they mapped these 7 purely shape-based territories back onto the brain, they matched up perfectly with different functional networks.
So let me get this straight. The shape of the receiver exactly matches the specific radio station it's tuning into.
Exactly. For example, the cells in territory MT1 receive uniquely different axonal inputs from the cortex, specifically somatic sensory motor inputs, compared to territory MT3, which receives associative input.
That is wild.
Yeah.
The physical form of the neighborhood perfectly predicts the type of information flowing into it.
It really does.
So quick recap again. They used 500-micron boxes, calculated these eigenmorph summary stats using WGCNA, and discovered 7 shape-based morphological territories that match up with distinct brain input networks.
Perfect summary.
Which brings up the ultimate biological chicken and egg question, and I want you listening to think about this. If the physical shape of a neuron perfectly predicts the kind of information it receives, does function follow form? Or does form follow function?
That is the million-dollar question.
It is, but honestly, that question becomes 1,000 times more urgent when we look at what happens when the system breaks down. Because mapping the perfectly healthy young adult brain, these postnatal day 56 mice we've been talking about, that's just step 1. The real power of this dendritome tool is seeing what goes wrong.
Right. To test the pipeline's diagnostic power, they compared those healthy young brains to 12-month-old wild-type mice to simulate normal aging. And they also looked at 12-month-old Q140 mice.
Q140, what's special about them?
The Q140 mice are a widely used genetic model for Huntington's disease. They contain the mutant Huntington gene, known as the MHTD gene, and specifically it has 140 CAG repeats, which triggers severe neurodegeneration.
Okay, so we have young healthy, and old with Huntington's. What does normal aging look like in this 3D forest?
In normal aging, when comparing the young mice to the 12-month-old healthy mice, they observed what we call dendritic atrophy.
Meaning shrinking.
Yes. It's a physical universal shrinking of the total length of the dendrites and a loss of the tiny branch tips. And notably, this withering happened pretty much equally across both D1 and D2 MSNs. Aging just universally shrinks the trees.
But when they looked at the Huntington's brains, it wasn't just accelerated aging, right? It wasn't just more shrinking.
Not at all. Huntington's disease caused highly unique, severe structural changes. At a whole striatum level, these Huntington's MSNs showed significantly lower complexity, fewer total branches. They also showed that higher centripetal bias we talked about earlier.
The echo chamber effect. The branches they did have were pulling inward, failing to reach out into the surrounding tissue.
Exactly. But here is where the regional zip code detail changes everything we know about neurodegeneration. Remember that rostral region at the front of the brain, the CPR, where D1 and D2 cells used to look similar?
Yeah, the front zip code.
Right. In normal aging, the healthy D1 MSNs in that specific region shrink, falling below the brain-wide median for length and complexity. But in the Huntington's mice, the D1 MSNs in that exact same rostral region stubbornly stayed above or near the median.
Wait, they didn't shrink when they were supposed to.
Exactly.
So the disease cells are actually bigger and more complex in this one specific area. That is the part that stops you in your tracks.
It really is counterintuitive.
Because Huntington's isn't just a universal dying off of the brain. The healthy cells prune themselves back as a normal part of aging, but the disease cells in this specific zip code are stuck. They are actively miswiring, retaining zombie branches they shouldn't have.
It's a pathological miswiring, yes. It's an inability of the neuron to adapt structurally over time rather than just simple cell death. And it is highly specific to the cell type and the zip code.
Okay, final recap before we wrap up. Normal aging physically shrinks dendrites across the board. But Huntington's disease causes specific, complex, structural glitches like zombie branches that vary wildly depending on the cell type, like D1 or D2, and the exact brain region, like the rostral zip code.
Spot on.
So think about this. If a disease like Huntington's damages different brain regions in entirely different ways, how should that change the way we think about designing a cure? Because if you're listening to this and assuming a pill just bathes your entire brain in a chemical fix, this study is saying that's nowhere near enough.
No, it's not. If we connect this to the bigger picture, the dendritome mapping approach isn't just a basic science achievement. It gives us an end-to-end systems biology tool for precision medicine.
Precision medicine.
Yes. By digitizing the physical shapes of neurons into a standardized 3D map, scientists around the world can now test a genetic therapy or a new drug and visually see at a massive scale if the physical architecture of the brain is healing.
That's incredible.
We can literally see if a drug fixes the specific structural glitch in the rostral D1 cells without inadvertently messing up the healthy cells next door.
It is absolute precision medicine, but for architecture instead of just chemistry. Which leaves you with this final thought to mull over today. We usually treat neurological diseases, from depression to Huntington's, with chemicals.
Right, usually.
We flood the entire brain with neurotransmitters like dopamine or serotonin, hoping to balance out a deficit. But if learning, aging, and disease fundamentally alter the physical, structural antennas of our cells, maybe the medicine of the future won't just be about chemical balance.
It's a profound shift in thinking.
Maybe it will be about structural landscaping, literally creating drugs that prune or fertilize specific microscopic branches in highly targeted zip codes of your brain's massive forests.
A whole new frontier.
It really is. Thank you for joining us on this deep dive. Keep questioning the shape of your own thoughts, and we will see you next time.